you here in this astute institution that I've heard about since just starting to get into deep sea biology with the Johnson Sea Links. Absolutely groundbreaking, and it's really a pleasure to come here and see what's still here and see that there's still research going on and a great program, and get to talk to people. So I work on primarily deep sea animals that don't live on the seafloor. I work on things that are in the midwater, we call it, which means anything below the surface of the ocean and up above the seafloor. As you can see from the animals I've shown here on my cover slide, they're generally fairly unusual animals. They don't look like the animals you're used to. Even if you were a marine invertebrate biologist, you would look at these and say, those are pretty weird. They're definitely strange animals in the deep sea. So the differences between pelagic organisms, which refers to anything that's up swimming in the water column, and benthic organisms, which refers to all the organisms that are living on the seafloor itself. You'll hear those two words a lot during this talk. Pelagic refers to the swimming things up in the water column, benthic on the seafloor. So here I showed several pairs of organisms with different examples. On the top here are forams. Those are single-celled amoeboid-like animals that submit shells. So here's a pelagic one, very spiny, long, cellular processes sticking out, very fine, very delicate shell. Benthic ones, considerably more substantial, little tanks, basically. Here you see a pair of nudibranchs. This is a typical intertidal, shallow one. This is a mesopelagic nudibranch. It looks like a jellyfish, basically. So you see a lot of transparency. A lot of their tissues get very watery, very gelatinous. There's you see where their appendages get very long. In the case of the forams, in these isopods here, this is one of the little roly-poly bugs that you find under logs. This is what they look like in the deep sea, up in the water column. Hyperid amphipods, transparent, amazing, strange eyes, unusual appendages, things like that. So the animals that live up in the water column, the pelagic animals, are very different from their benthic ancestors. And most of the pelagic groups of animals that we have today originally evolved from benthic ancestors. So they are relatives, and their closest relatives and their ancestors were on the seafloor, and at various times they've moved up into the water column. And by studying how those animals are different from their closest relatives, we can start to understand what they're capable of in an evolutionary sense. So in general, I'm interested in looking at the diversity of invertebrates out there in the pelagic realm, out there in the midwater. I want to know who's out there, what they're doing, how they've adapted to this strange habitat. In my research program, my general objective is to contribute to our knowledge of pelagic invertebrate community composition and evolution. And I do that with three-pronged approach. Basically, the basis of all my projects are rooted in natural history. What are the animals? What are they doing? How are they related to each other? You can see that in two examples of projects. This project up in the top right corner was a description of a pelagic chaetopterid worm. Chaetopterid worms typically live in tubes attached to the bottom. They're really interesting worms on their own. We found this ping pong ball up in the water column at about 1,000 meters. We kept finding them. We're wondering what exactly they were. They look a lot like a larva of a chaetopterid. They're just 20 times bigger than they're supposed to be. So they have a lot of characteristics of chaetopterids and stuff, so describing that animal was really interesting. Basically what happened is they've decided not to continue growing up, and they maintain a bunch of their larval characteristics, and they just stay up in the larval habitat and never come down. Another example of a natural history project is looking at diversity within a single species. So most of the taxonomy that's done in the past on these isopods that I worked on for my dissertation at Berkeley, most of the taxonomy that's been done has been based on morphology, and there's very specific sets of characteristics or specific sets of features that these animals have that they use to just distinguish between different species. What I found within this single species is this is the head of a male or a juvenile female or a juvenile. They have these incredibly large jaws. Instead of their jaws functioning like this, like a normal isopod, they've actually moved the jaws out, and they now work like this, and they have these gigantic muscles in the head that they use to function. When the animals brood, when a female broods or molts into the brooding stage, where they are carrying their young around, they completely transform the entire shape of their head. They get rid of those huge jaws. They have normal little isopod jaws that kind of sit here and shred things up. And so those characteristics alone would normally have distinguished, at least a species, if not a higher taxa. But within a single species, we found that kind of variation. So those are the types of natural history projects that I work on. Evolutionary biology forms the framework and a lot of the interesting questions for what I do. A lot of phylogenetics, where we look at how things are related, what the evolutionary history of things was, what the evolutionary history of diversification within a group is. So here's an example of that, the isopods, those isopods that you saw up in the top left corner in the last slide. This is a tree diagram that explains how they diverged from each other. And that is a type of phylogenetic project. I'll talk about some of those later. Also work on starting to get into population genetics, where we look at, within a single population, how are all the individuals related to each other. And this is, we call it a haplotype network. It basically tells you how each of the different individuals are related, how much interconnectedness is there between those dots, tells us how the population is functioning and how far it's spread out. And then finally, marine ecology forms a big part of my projects as well. And some examples of that are to look at the distribution and the population dynamics through time of these pelagic forams. Was a really fun side project when I was at Scripps. I got to talking with some paleooceanographers. They were really interested in learning about what live forams do, because they always study dead ones and they assume that they can translate what they were doing based on the little bit of information that they have left over from the fossils. We see these live ones with remotely operated vehicles all the time, so they got really excited and wanted to know a little bit more about what they were doing. We started looking at them and found out that they occur in a very thin layer at about 300 meters, and basically they're a little minefield for the ostracods and copepods that are swimming down during the day to hide out in the oxygen minimum zone and then migrating up to feed at night. And those forams eat those multicellular animals. Those singular cellular forams eat the multicellular animals that are migrating back and forth past them through this thick minefield of forams. So that was a fun side project. And then I've looked at feeding ecology of various isopods and some of the polychaetes. So tonight, what I'm going to talk to you about, I'm going to introduce you to the pelagic realm and talk a little bit about my perspective of how the animals that are living there, what are some of the challenges that they have to deal with in that habitat. It's a very unique habitat. I'll talk a little bit about the tools that I generally use for this type of research and why it's particularly timely to be working on pelagic biology. And then I'll talk about two small projects dealing with two different groups of animals, serratulariform polychaetes and acorn worms. And then we'll summarize everything by looking back at the habitat and the characteristics of that habitat and seeing what we can learn from the biology of those animals and what we can say about pelagic adaptation. So the pelagic realm, or the midwater, is, again, everything below the surface and up above the seafloor. When you look at this video, if it was a little darker, it's okay. It basically looks like an 80s screensaver. Like you're flying through space, all these things are heading towards you. Periodically, as you get used to watching the video, you see animals pop up. They're big, large animals that you can recognize. The more of this you watch, the more animals you can pull out and identify, the better you get at identifying the different animals. Most of the particles that you're seeing there are marine snow, that's phytoplankton that is sinking down, that's dead animals, fecal pellets, all kinds of organic matter that's sinking at various rates down through the water column. And then there's all of these animals in there. So the pelagic realm is huge, right? 1% of the Earth is covered in oceans. 90% of that is more than 200 meters deep. And that accounts for about 99% of the living space on Earth. So the midwater, or the pelagic realm, is the largest habitat on Earth by volume. And we know very little about it because it's very difficult to get to. It's huge, which means there's a lot of space, a lot of volume to explore. There's also a lot of space and a lot of volume for things to be spread out in. It's dark. Once you get below about 200 meters, there's very little light. Once you get below 1,000 meters, there is no light, even in some of the clearest water. So it's very dark. Most of the light that is in the areas of the water column that I work on is produced by bioluminescence. It's produced by the animals that are living there. It's also, the pelagic realm is three-dimensional. We live in a three-dimensional world, but everything is tied to a surface. So it's more of it's kind of a three-dimensional world that's flattened down and associated with a surface. In the midwater, there are no surfaces other than other animals or things for things to live on. So they really are truly in a three-dimensional habitat. They're having to look for food and look out for predators below them, above them, from the sides, from the back. There's nothing to hide behind except for other animals. So it's a real challenge for camouflage or for finding food. This diagram gives you an idea of the different habitats by volume and by depth. So the brown stripe basically is the benthic habitat. All the blue is the pelagic habitats, and terrestrial is up there. So the pelagic realm is also, it could be described as syrupy. The animals that live there are anywhere from a few microns to 50 meters in length, 150 feet. If you watch the video of this little isopod, you see it swimming and it's pulling itself through the water. As soon as it stops pulling itself through the water with its appendages, it stops immediately. So it is essentially living in syrup or in honey. The majority of the animals out there are experiencing the water column much differently than we experience it when we're in there, and we can swim around quite easily. It's quite fluid. The dissolution rates and the ability to move in that fluid is very different for most of the animals that are there. It's also very patchy. So it's much more common to encounter a couple of the same types of animals within close proximity and not again for a long time. So they have to be able to find mates. They have to be able to find prey in this very large, very spread out, very patchy habitat. And it's highly structured in layers. So if we superimpose on this little diagram a few different layers of water movement, most of the water out there is moving in horizontal sheets, and those sheets can be moving at different velocities. Those currents can be different strengths. Basically what it sets up is this huge layer cake effect. And so it's much more common to find the same organism at the same depth, very far apart biogeographically, but at different depths in the same place, you find different animals. Okay, so it's all kind of this layer cake, and things are living in those layers. Animals, midwater animals, usually have very distinct depth distributions, and so we'll talk a little bit more about those in the future. Here you can see an example of some of that layering. These are water column profiles. Green is salinity, yellow is particles that are in the water measured by transmissometry, and purple is temperature. And even at this very rough scale, we're only looking at 1,500 meters. At the top there, you see evidence of layering in the water column. It also is variable through time. So this is 11 years of summarizing oxygen concentration you see in the summers. Oxygen is considerably lower at this depth than it is in the winters, and that is a typical feature. So there is seasonal signals, even in the very deep sea. There's fluxes of phytoplankton and productivity from the surface that make it into the deep sea very quickly, and so there's seasonal signals that we find in the deep sea, and there's variability through time. So all of these characteristics of the pelagic realm set up a very unique habitat that these animals are trying to adjust to and trying to survive in. And that is probably what's driving all of these strange morphologies and changes in behavior that we see in pelagic animals. And so I'm interested in those adaptations. What does it take to survive up in the water column? Once your group of animals invades the water column and starts trying to survive there, how does their morphology change? How does their behavior change? So before we go off this slide, this is kind of a diagram of the midwater. This is the top 200 meters. It's referred to as the epipelagic zone. We know quite a bit about the epipelagic zone, and we know quite a bit about the fishes and other hard-bodied animals that are down in the pelagic zone, because we've been trawling, putting nets in the water, and pulling specimens up for the last 200 years. There is a wealth of diversity out there that is delicate, that's gelatinous, that we haven't been able to sample. And so that is primarily what I focus on. Most of the work that I do is focused on those delicate animals because it's the things we don't know anything about. I find that very exciting. Most of the work that I've done in the deep sea, midwater stuff, has been done with the Monterey Bay Aquarium Research Institute, which has 2 ships and 2 remotely operated vehicles. They essentially have the same capabilities as the Johnson Sea Link, except they're not quite as fun because you don't get to go down in them. But we shared a lot of technology. So the suction sampler that you guys developed here, we use there on the vehicles. The detritus samplers that were developed here, we use there on the vehicle. So really very nice complementary systems, and I'm just really sad to have seen the Sea Link go into storage because it's a great system to work with. This is kind of a parallel system, but without people in it. This is a remotely operated vehicle. It is tethered to the ship, operating ship at all times. We sit in a control room in front of a wall of monitors, watching what the vehicle is seeing and controlling it, flying it around, deciding what we want to look at, what we want to pick up, what we want to do. It's outfitted with about 14 different cameras, a high-definition camera. It's outfitted with collection devices like the suction sampler with detritus samplers. Detritus samplers are really fabulous for midwater. They essentially make it so we can collect an animal without it even knowing that it's been collected. So it's a canister about this big. The top and the bottom slide open. Basically, we see an animal we want to collect. We fly this large vehicle about the size of a VW bus or a little bit bigger. We fly it up to the animal, put the canister over it, close the top and the bottom. And you bring the animal back in the water that it was living in until it bumps into the sides. It never knows it was even collected. They come up in really fabulous shape. And that is really unheard of for midwater animals. You can collect ctenophores, you can collect all these incredibly delicate animals with this. Oh. There's also a suite of sensors on there, so we can tell exactly where we're at geographically. We can measure oxygen, temperature, salinity, all kinds of things like that right there. So we can really hone in on, okay, where are these animals occurring and what are the characteristics that they're experiencing, which we couldn't do before. Even with nets, we had CTDs and stuff on them. You never knew exactly where you were getting the animal and what part of that track, matched up with what they were seeing. Now we can really pinpoint that information. And MBARI, basically, whenever the sub is in the water, the videos are recording. They have 25 years of archives now, archived oceanographic data to go with that. It's all time code linked back. It's wonderful. So why are the ROVs so useful? We can start now to understand ecological processes in the midwater. For example, these are the discarded houses of a larvation. It looks like a little transparent tadpole. They are these little tadpole animals. They make these really elaborate mucus filters. They have 2 layers, a very specifically shaped inner filter that they use to collect food, and a big outer filter that they use to keep out the bigger particles that they don't want to have anything to do with. On those outer filters and on some of the inner filter is a collection of all kinds of organisms that are using that as substrate, and a collection of all of these particles that they've filtered out. They produce about 3 of these per day, and then they discard them and build another one once they're clogged. And once they are no longer pumping water through that filter, the filter collapses and sinks very quickly to the deep sea. So for a long time, they were trying to measure how much carbon was going from the surface productivity into the deep sea, and there was this huge hole in the equation where they couldn't ever figure out where the extra carbon was coming from to support all the animals that were down there. And as it turns out, these very large, very ephemeral, floaty particles never, ever get collected in sediment traps, which is the primary way of measuring how much carbon is being transported through the water column. And so with ROV, we were able to visualize these, count them, collect them, measure the amount of carbon that was in them, measure their sinking rate. And in 2005, my supervisor at MBARI did this wonderful paper where they filled in this hole. And so that's the kind of thing that we can do with remotely operated vehicles or with submersibles that we couldn't do before. Also, we're able to look at all of these very delicate, strange animals and start to get a better handle on the biodiversity of animals that are out there, fill in our gaps of knowledge. And now that we're starting to understand the ecological processes and the biodiversity that's out there, I think it's perfect time to start looking at the ecological or the evolutionary processes that are taking place in the midwater, now that we can really start to get a handle on that habitat. And so that's where the next tool that makes midwater biology comes in very handy. So genetic tools help us with a major problem that we've had in deep sea biology and pelagic biology for a long time. Ernst Mayr, a very famous biologist, once pointed out that being able to describe the variability within a species is a major problem for trying to look at biogeography or trying to look at ecology, trying to look at different aspects of taxonomy. Genetic tools now allow us to match up different life stages. In pelagic biology or in deep sea biology, you often only have one specimen from one place, one specimen from another ocean basin. Are they similar enough to be the same thing? Are they parts of the same animal, different life stages? You don't know when you only have just a couple. You need a whole bunch of samples to fill in all the gaps in between that. With genetics, we can actually match those guys up. Are they the same thing? So we can match up life stages. We can identify cryptic species where the morphology hasn't changed, but the species have diverged in different locations. We can look at variation within a single species, not having to have a million specimens of something, just the couple specimens that we can get. And so now we can really start to get an idea of how much biodiversity is out there, what are we missing with genetic tools. So it makes it really, really important. Traditional tools are still incredibly important. My favorite thing to do is sit at the microscope or sit at sea and take pictures or look at these animals, these incredible animals, and look at their morphology, look at how they've changed, how they're functioning in their habitat. So it's still very important to do the basic observational descriptive work that people have been doing for 200 years. So I say that I do very traditional biology with very cutting edge tools. But very, very basic stuff, a lot of it. So now I want to tell you about a couple of the projects that I've worked on. And the first is on serratuliform polychaetes. When I was a graduate student, midwater biology is very opportunistic. You go out, you never know what you're going to find. It makes it a difficult thing to do because you go out, the submersible is down for 12 hours. You don't want to leave the control room because you never know what's going to pop up. And so it makes for some very long days because you spend 12 hours in the control room and then you spend another 12 hours processing the animals that came up and looking at them and studying them. As a graduate student, I was out there collecting isopods, and we kept finding these strange, beautiful worms down deep. And Steve Haddock, a biologist at MBARI, was very interested in bioluminescence. He started collecting them because they have these fantastic little capsule-like structures right behind their heads. And when you disturb them or when you pinch them in the lab, they pop off one of these structures. And as soon as it pops off, it turns bright green, glowing, and the animal swims as fast as it can backwards. And so he was really interested in this bioluminescence because it's a different group of worms that they hadn't seen bioluminescence in before, and he was interested in the chemistry and the behavior associated with it. So he kept collecting these things, and he kept giving them to me because I was the polychaete person at MBARI, and saying, what are they? And I was saying, I don't know. I don't know what those are. They're really weird. You know, they have a few characteristics of this group and a few characteristics of that group. There's nothing like them described. And as we kept collecting them, we kept finding more and more. So he found the first species. I found 2 more species just in Monterey Bay. Fred Plale was a collaborator who came out on another MBARI cruise. He found another 2 species up off of Oregon. Larry Maiden, a researcher at Woods Hole, was doing a research expedition in the Celebes Sea off Indonesia and found this fabulous animal. They called it squidworm because it kind of looks like a squid and a worm fighting or something going on here. These are really large animals. They're about this big. That's the body of the worm. Once you put those head appendages on, they're huge. And they were everywhere down there. James Cameron actually just saw some on his expedition recently. There's been a couple other expeditions to around Indonesia, and they've all recorded these guys. So they're
Very abundant, they're large, they're interesting. Nobody had ever recorded them before or described them before. Turns out they're closely related to the bomber worms that I'd been collecting off of Monterey. They also found a second species of bombers in Indonesia. So Greg Rouse, a researcher at Scripps Institution of Oceanography, really fabulous polychaete person, was willing to let me come to Scripps and work on this as a postdoc. Once I finished my dissertation on isopods, I switched over to polychaetes and finally got a chance to work on these guys. Wanted to figure out where exactly they went, what kinds of polychaetes they were. They shared the most morphological characteristics with this group of polychaetes called Acroceridae. Very boring group of polychaetes. Small, thin, on the bottom, very difficult to tell apart, probably about 15 species described in 3 different genera. Really boring group, but they share some of the same morphology of their chaetae and of their head appendages. And then there's this group, Pelagic, and they are very diverse. Some of them have this thick gelatinous sheath, which is common in the pelagic ones, have a gelatinous sheath as well. They share some similarities of head appendages. There's already 2 known groups of Pelagic that are pelagic. This is the swimming Vienna sausage Flota, Vitjatsi. There's 3 species described. It's a pretty fabulous worm. It also bioluminesces. Really interestingly, it doesn't spew off any parts or anything when it glows, but it does produce some really nice, beautiful green bioluminescence at the base of those parapodia that it's swimming with. There's also Piobius, which is probably the most abundant pelagic invertebrate off the coast of California. We went back and looked through the archives, saw 133,000 observations of these animals in the last 10 years. They're super abundant, and they occur through the water column all the way from the surface down to 3,000 meters, but that's another project I don't have time to talk about. So basically what I did for my postdoc, there's super tiny writing on here that you don't need to read. Basically, I sampled as broadly as I could through these 2 families of polychaetes and all the worms that I had collected so far. Created a morphological matrix. In other words, what are the characteristics of these different animals and how do they relate to each other? Looked at 5 different genes for them and vouchered all the material. When you're doing midwater work, it's really important to voucher everything that you have because your identifications are always a little bit iffy because you don't know how widespread different species are. So this was the result that we got trying to figure out where these animals go. And what you're seeing here is a phylogenetic tree. Basically, a phylogenetic tree helps us understand the evolutionary history of diversification of a group. And so you can follow these lines and see, okay, so they split here. Here's the Ceratulidae. Here's the Pelagic and Acroceridae. We call these sister groups because they split off from each other. And this just tells us, okay, so these guys are more closely related to each other than this to this, right? So it's just a tree. It's a way of visualizing the history of their relationships. What we found is that all of the bomber worms and the squid worms were in a single group. They're all more closely related to each other than to any other known polychaetes, and that they were within the Acroceridae, so that little boring group of worms is what they belong in. And so we didn't have to name a new family, but we have like 4 different genera that need to be named and about 9 species. I had found 7 by the time I did the postdoc, and then actually in the last 2 months we just found 2 more species. So it's ongoing as we keep exploring further. What I also found was interesting about this project is, well, let's go to the next way of looking at this. So here's the Pelagic. These are all those other worms. Here's Flota. Here's Piobius. The interesting thing about this phylogeny is that you have a pelagic origination here, and then you have all of these guys, which are all the new pelagic guys evolving from there. This is an easier way to look at it. So everything in bright blue here are the pelagic things, and they're spread all over the tree. As an evolutionary biologist, this is really exciting to me because it gives me replicate of 3 times that things have evolved from benthic ancestors into pelagic things. So that's 3 different ways of seeing how that transition happens. So We'll talk a little bit more about that at the very end of the project. Just to summarize the polychaete section, so we discovered a new novel bioluminescent structures and behaviors associated with that. We're describing a new clade with 9 new species, a couple new genera, and now we have a well-resolved phylogeny or evolutionary history for the Pelagic and the Acroceridae, and that allowed us to place the new species and name them correctly, and to identify several cryptic species that supposedly within the Pelagic there's these species that were really, really widespread, and their morphology was not different enough to be able to distinguish them, but genetically incredibly distinct, and so there's definitely some taxonomic work that needs to happen there. And then we have 3 examples of pelagic lifestyle arising within this, these 2 groups. So now I want to talk about acorn worms. So the polychaetes are annelids, like segmented worms. This is a completely different group of worms, but they're still worms, so they're within the realm of interest. This is my introduction to them. We were looking for isopods, and we found this creature floating along just off the surface of the seafloor. It's actually easier to see its shadow here till we really zoom in closer. Basically, it's an orange, about a foot long, about an inch wide worm with very few features. Really very strange thing, not something that anybody on board was familiar with. So we collected it because we wanted to see what it was. Brought it on board, looked at it, decided it was an acorn worm, a very strange one. A couple months later, we found this animal floating about 20 meters up off of the seafloor. This guy is also about the same size, just shy of 1 meter or 1 foot long. Pretty large animal floating away. Up there we collected, no, they didn't collect that. I wasn't there. They didn't collect it. They looked at it, took great video, and then drove away. Anyway, so I gave the specimen that we collected that I was there for, I put it in a jar. I said, okay, I'll put it away when I teach invertebrate biology at some point. I'll show it to my students. It'll be a really cool thing that they wouldn't have seen anyplace else. I went down to Scripps, and actually 2 doors down from me was Nick Holland, who is the only one who's ever described a deep-sea acorn worm in recent history, who's still alive. And I said, oh, you know what? I have this specimen. Let me show it to you. I gave it to him. He got super excited. I mean, he's a dramatic, wonderfully, theatrical person anyway, but he was super excited about this worm because he had never seen one of these deep-sea animals. They are so delicate and so fragile that the one he had described before was in 4 pieces. The one I gave him was this long and was in 1 piece, and it was beautiful. He could see all the features on it and stuff, and so he was really excited. I said, okay, great, take it, describe it, do whatever you want to do with it. I was just going to show it to my students, but if it can be used for, science in another way, great. So that's how I got involved with the acorn worm project, even though they're variably benthic and just starting to make that transition into the water column. So acorn worms belong to the phylum Hemichordata. Hemichordates are deuterostomes along with echinoderms or sea urchins and sea stars and things. Funny little worms called xenoturbella, hemichordates and chordates. So they all belong in the single group called the deuterostomes. And hemichordates are thought to be really important. There's a lot of research that goes on about them because they are basal to chordates. And so understanding how the chordates arose, you have to understand where they came from. And so a lot of people work on hemichordates because they're interested in that. The hemichordates consist of 2 groups, the Enteropneusta, the acorn worms, which you see a typical shallow water representative of here, and the pterobranchs, which are little colonial branching critters that there's only a few species of known. So acorn worms, their bodies are basically broken into 3 sections. You have the trunk of the body, which is basically where the gut and the reproductive organs and things are. You have this narrow collar, which forms the back of the mouth, and then you have a proboscis that they use to dig with, and it forms the front of the mouth. So they're very simple body plans. They do have something that is thought to be like a notochord. Within the acorn worms, before we started working, there was about 89 species described. They are pretty much all benthic. They pretty much all live in burrows that they dig. primarily in shallow water. There's 4 exceptions before we started working of acorn worms that were found in the deep sea. So one was found at vents when they first discovered vents. Nick had described this one other deep sea species, and then there's another one that Nick had described fairly recently, and one that was described from Challenger material a long, long time ago. There's basically 3 families known: Sphingalidae, Tychodaridae, and Herimanidae. And then there's this weird larvae form that when it was first found, they're very uncommon. They're typically found way out in the middle of the ocean. They are very large. This is almost 3 centimeters, so just over an inch in size. Really very strange. They were originally described as their own phylum, which, you know, whatever. Anyway, finally somebody realized that they were actually a larval form of these acorn worms, but they've never found out who the adult is. But there was some suggestion that since they're found way out in the middle of the ocean and there's so few of them, that probably they were from some abyssal acorn worm or something, but they didn't know what abyssal acorn worm because they didn't know about any abyssal acorn worms. But anyway, so there's Planctosphaera pelagica in this group, but they don't know where it goes. So what did we find? So I gave Nick the specimen. He sectioned it. He, started describing it. I said, well, if we're going to do that, we should go back through the database and see how many other acorn worms we've seen in the deep sea off of California, because I know we see them fairly often. This is a little summary of some of the different shapes and sizes and colors that they come in. They are very common in the deep sea. We had about 500 observations with MBARI vehicles just off of California. Once we started working on that project, somebody, Monty Preed from University of Aberdeen, was working on the Mid-Atlantic Ridge, and the primary animals that they were finding out there were also Enteropneusta, like this species right over here. So they collected a few and sent them to us to include in our project. They typically occur, the shallowest one we've ever found is 1,600 meters, and they go down as deep as we've really looked. They're diverse, so there's 13 species still undescribed and 4 species that we've described so far. And everywhere we look, we find new ones. A few years ago, in 2003, we went to the Gulf of California. We found 2 species. We identified them on video. We just went back to the Gulf of California last month, and we found one of the species we saw there before, but then we didn't find the second one, but we found a different species, and it's all over the place. So they're very diverse. As we start exploring the deep sea more, we'll probably find more. And they're epibenthic. They don't burrow in the sediment. They live out on the sea surface, and they basically graze along and clear that top layer of the most freshly settled mud that's down there. They are also drifters. So the first one that I saw drifting along above the sea surface, that is a very unusual behavior for an acorn worm. They are supposed to live down in the sediment in their tubes and not leave. In the deep sea, evidently, it's advantageous to get up, move around. The way that they seem to do that, their bodies are very gelatinous, very fragile. They empty their guts, so they use their gut content, which is all sediment. They empty their gut contents, so they're dropping their ballast, essentially. They lift the anterior half of the body up off the seafloor, and they produce a mucus balloon. And evidently, that creates enough drag to pull them up into the seafloor currents. They coil into a very tight, very specific shape like this, and then they drift along. When they're ready to settle back down, they seem to uncoil, make their bodies very straight, and they settle eventually back down onto the seafloor. Pretty amazing animals. And that's really the only link. They're just starting to use the water column. So, give them another 100 million years, we might have a fully pelagic one. That would be great. Also tissue sampled most of these things, everyone that we could get tissue for, and we're able to start to get an idea what the evolutionary history of the hemichordates in general are and where all of these new ones occur. So here's Herimanids. This is one of the other families that's established. Sphingalids, another family. Tychodaridae is where we thought they belonged, but they actually are their own family. Nick named it Torquoratoridae. That's the only time I'm going to say it. It's unpronounceable, but it was appropriate for the first one that he saw, very descriptive. So what we found is 5 different genera that we're in the process of describing. When we have really good specimens for them, we can describe them. The molecular work has just been absolutely beautiful. It fits perfectly with the morphology work. It's well supported. If you read these support values, it shows you how much we believe in each of those splits. So it's really been a pleasure to work on this project. There's morphological characters that support it. So all of these guys have no proboscis skeleton. So in that front part of the body, they usually have a skeleton. In other acorn worms that look something like this. But in these guys, they either have no skeleton or they have this very tiny little plate that's a leftover piece of that skeleton. These pictures show you some of the different shapes that they come in. So we can identify genera on video. We can't identify species on video necessarily, but we can identify the different genera on video, and their morphology really fits well with that. In the process of describing all of these guys, we found some really interesting things about their biology. We found a few examples that are hermaphrodites, so they have both female ovaries and testes as well, so they make both eggs and sperm, and they seem to mature at the same time. So there's some question, is that something that they have to do to be able to live in the deep sea where it's hard to find other individuals, or when you find another individual, you can always mate with them because they've got both? So a few examples are hermaphrodites, which have never been found before in any acorn worms. They brood their young. This is brand new information. Here you can see a little larvae. If you pull open these flaps, we call them the gonadal wings. If you pull those open, the inside of that tissue is just packed with eggs and with developing larvae. And they also have these incredibly large externalized eggs, some of the species. So this is looking at the epidermal wall. This is the body wall of this tissue here. And you can see that there's a very thin layer of epidermis membrane and a little stalk that attaches it. These eggs are actually outside of the body. So they're hanging their eggs just on the outside of the body. What is the purpose of that? We don't know yet. It's something that we're interested in looking at. Here you can see what they actually look like. You see all these eggs poking out here. And the eggs themselves are gigantic. They're 1.5 millimeters in size. So it's not the largest invertebrate eggs. Cephalopods have some, gigantic 4-centimeter eggs. But as far as other than cephalopods, these are gigantic eggs. So what's going on with them? Very strange. Are those gigantic eggs leading to Planctosphaera larvae? Maybe. We don't know. We need to collect some of these guys so that we can match them up with genetics and see what they turn into, because it isn't really possible to raise these guys and find out what they grow up into, but genetically we can match them up. So it would be nice to be able to solve this question. So in summary, acorn worms in the deep sea are very diverse. We have 9 species from the Northeast Pacific, 2 species from the Celebes Sea, several other species from other places. They only occur below 1,600 meters. They're exclusively deep sea. They drift in demersal or deep sea bottom currents. They have very unusual reproductive biology. We found several different examples in different groups. The phylogeny that we now have provides a framework for describing the new genera and the new species. Unfortunately, because these worms are so delicate, it's very difficult to get a good specimen, and you don't want to describe it unless you have a really good specimen. So even though we've found about 19 species, we only have good specimens for about 10 so far. So we're working on the descriptions for those. And so this is a group that we can see in the phylogeny originated in shallow water. All its relatives are shallow water. The first ones they've spread into deep water. The interesting thing about that is now they're exclusively deep sea. There's no leftover relatives up in shallow water within this family. So they're kind of an unusual example. So to finish up here, I just want to take you guys back to the beginning of the talk and look at, again, the characteristics of the water column of the pelagic realm and see how that relates to animals, how they function. So the pelagic realm is very light limited, and that has led to many adaptations, such as bioluminescent. Most pelagic things are bioluminescent in one way or another. or they're using bioluminescence somehow. Bioluminescence is really common. Transparency, variations in their pigmentation and reflectance. There's a lot of interesting research that's been done looking at how animals hide, either by covering parts of their body, changing the shape of their shadow with pigmentation, covering with bioluminescence, different things. Lots of work has been done on that. Changing their shape, so that nudibranch that I showed you guys earlier, the pelagic one, it no longer has a shadow of a nudibranch. It doesn't swim around like a nudibranch. It looks like a jellyfish. So a fish or something swimming around says, oh, I don't want to eat that. That's a stingy, yucky thing from far away. So a lot of animals have changed their shape to hide what they really are. And then sensory development, there's lots of examples of, particularly like in the hyperid amphipods, these really elaborate eyes that they have to be able to pick up just the tiny, tiny amounts of light that are there, or to be able to see different types of bioluminescence, or use bioluminescence to see things that they haven't seen. But I'm sure you guys have had talks from Tammy and Edie about those types of things. Because all the animals in the midwater have very specific depth distributions, somehow they have to detect where they want to be, what is the optimal area for them. And then they have to somehow be able to maintain their position in that water, which means they either have to be buoyant or they have to swim all the time. They have to do something to maintain their position there. As far as I know, there's been zero research, and I would love to hear about it if anyone knows, about how midwater animals detect the characteristics of the habitat that they're in, or the depth, you know. How are they figuring out where they want to be? How are they monitoring that? How do they know when they've gotten out of conditions that are survivable for them? There has been a lot of work on how things maintain their position in the water column, with looking at different ways that they manage their buoyancy, either by accumulating light ions or lipids, or increasing their surface area so that they sink slower. Lots of different ways that people have looked at that. Because the pelagic realm is so large, the food and the mates tend to be really spread out or to be really patchy, so they're hard to find. So I would expect to find lots of adaptations in their sensory development for being able to detect their food or detect their mates in this habitat and find them. And then finally, all three of those characteristics of the pelagic realm have led to food limitation. We see lots of adaptations to being able to find food, or being able to make the most of food when you find it, like by handling very large prey items, making very inexpensive watery or gelatinous tissue, relative inactivity, so they don't do a lot of swimming during their lives, or being opportunistic generalists that can eat anything and everything that they happen to come along with. So I just want to give you one example of the types of things we can do with, when you have an evolutionary history, a good phylogenetic hypothesis, and the types of questions we can ask. So back to the tree of the ceratuliform polychaetes. Remember, we have these three examples of pelagic life having arisen. So we can ask questions like, is being gelatinous an adaptation to pelagic life? And if we highlight in purple all the things that are gelatinous, we see that here's the pelagic things, but here's a bunch of non-pelagic things, a bunch of benthic things that are also gelatinous. So no, being gelatinous is not an adaptation to pelagic life, but what we can learn from this is that being gelatinous may be a way to facilitate moving into the water column. So within this group of animals where making gelatin is a fairly common thing, maybe they can then co-opt that ability to be able to move into the water column and have done it multiple times. So this just leads us to more questions. What happens to that gelatin once they move up there? Do they change the structure of it? You know, how do they use it? Do they produce more of it? Things like that. Another way that we can look at things, and this is a much earlier stage, is looking at sensory development, and that feeds into looking for mates, finding food, things like that. This is a benthic acrocerid. I don't know if you guys can see it from here, but on its head it has this squiggly little yellow line, and that is its nose. It's basically a ridge of ciliated cells with chemosensory cells in it, so that's its nose. It's fairly large for a polychaete. It's got a pretty big nose. If we look, this is a benthic one. If we look at a pelagic one's nose, this is what we find, a bush, basically. If we pull out one of those structures, we see that each of these, there's about 32 of these structures on this species. If we pull out an individual one, we see that that little yellow ridge of ciliated chemosensory cells goes up on that freestanding structure and goes out on each one of those little branches. So they have a huge sensitive nose, tons of chemosensory cells compared to the benthic ones. And they've started moving those sensory cells out away from their body, out into the water column so that they can detect things from further away. An increased range for their sensors we see in squidworm as well. These are their branchiae that they used to breathe with, actually, but on the very tips, they're no longer used for picking up oxygen on the very tips. The tips are highly innervated and extensible, and they're actually chemosensors and mechanoreceptors out there. And when these guys are undisturbed, those little filaments are spread way out, far away from the body, and they hold those branchiae in a very specific position that basically creates a sphere around them. So they have a burglar alarm system, essentially. Is anything coming near me? Can I smell something? Can I feel something coming at me? And so this is something that we see in a lot of pelagic animals where they've got very long appendages, they've got sensory structures way out, far away from their body, so they have a little bit of a heads up on who's coming at them or what's around them. So this work in midwater biology in general is very collaborative. Anytime anybody gets ship time, we all try to share it and help each other out when we can, because it's hard to get out there. It's expensive to get out there and get these animals and look at them. So I have a ton of people to thank for invitations on cruises and help in the lab and bouncing ideas off and stuff. So in general, couldn't have done any of this without these people. So that's it. If you have questions, I would love to take them.